Hello everyone, I'm Jennifer Nguyen from the University of Toledo College of Medicine and Life Sciences, Department of Neurosciences. I will be discussing my poster, Developmental Pyruvate Exposure Disrupts Molecular Pathways for Circadian Rhythms and MAP Kinase in Mouse Brain. Neurodevelopmental disorders are a category of pervasive disorders of the developing nervous system with few or no recognized biomarkers. This incidence of neurodevelopmental disorders is rapidly rising, with 17% of children in the US now affected. A significant portion of the risk for neurodevelopmental disorders, including attention deficit hyperactivity disorder, is contributed by the environment, and exposure to pyruvate pesticides during pregnancy has been identified as a potential risk factor for neurodevelopmental disorders in the unborn child. Developmental pyruvate exposure in mice results in an increase in dopamine transporter that directly causes an ADHD-like behavioral phenotype in mouse. My experimental question is, does developmental exposure to pyruvates cause other brain-wide molecular changes? Here, we use an integrated multi-omics approach to determine the broadest possible set of biological changes in the mouse brain caused by developmental pyruvate exposure. Using a litter-based split sample design, we exposed mouse dams during pregnancy and lactation to the pyruvate deltamethrin at 3 mg per kilogram every 3 days, a concentration well below the EPA-determined benchmark dose used for regulatory guidance. We raised male offspring to adulthood, euthanized them, and pulverized and divided whole brain samples for split sample transcriptomics, kinomics, and multi-omics integration. We wanted to identify changes in gene expression caused by developmental exposure to deltamethrin. From our transcriptomics analysis, we performed differential expression of genes analysis. Figure 1a is a volcano plot showing PIR2 and CRT as significantly differentially expressed. These two genes are circadian clock genes, which serve as the molecular basis for animals' 24-hour internal timekeeping. Then, we identified genes of interest. We performed variance partition and selected the genes with a variance greater than 0.15 and a p-value less than 0.05 uncorrected. This resulted in 65 genes of interest, which were used for gene set enrichment analysis. Gene set enrichment analysis showed that genes encoding for circadian rhythms were enriched among our genes of interest, shown in Figure 1b. Next, we wanted to identify the transcription factors and kinases upstream of genes of interest. We used CHIA3 for transcription factor enrichment analysis. Results are shown in Table 1a. We used KIA3 for kinase enrichment analysis. Results shown in Table 1b. Genes encoding for transcription factors and kinases involved in regulation of synaptic plasticity, adult neurogenesis, and circadian rhythm were overrepresented among our genes of interest. Casein kinase 2A1 regulates all three. Next, we wanted to identify changes in kinase activity caused by developmental exposure to deltamethrin. We used a PAM gene chip with an array of kinase substrates to measure kinase activity in our samples, and we calculated a z-score representing differential activity between groups. Figure 2a shows 7 kinases of interest with z-scores above 1.5, all of which are involved in synaptic plasticity. Figure 2b shows the activity level of the top 10 kinases, and as you can see, 9 of the 10 kinases have increased activity levels. Figure 2c is a combination of 4 different analyses: KUSA, KIA3, PTMC, and UCA. MAP kinase 3, also known as ERK1, and RAF1, also known as CRAF, were present in all 4 analyses with high confidence. Lastly, we performed multi-omics by integrating our transcriptomics and kinomics data to identify multimodal changes in molecular pathways. Kinograte and PAVR were used for fast gene set enrichment analysis and clustering of the pathways. Figure 3a shows 13 significantly clustered pathways. Regulation of MAP kinase cascade was the top of the list, followed by regulation of transferase activity, aka kinase, and then apoptotic processes. 3b is a protein-protein interaction network with MAP kinase 3, also known as ERK1, at the center. Through multi-omics, we were able to identify changes in molecular pathways for circadian rhythms, MAP kinase, growth apoptosis, and synapse function, shown in Figure 4. For the future, we have repeated this experiment on 112 prairie voles, and we dissected the suprachiasmatic nucleus, the brain region which controls circadian rhythm. these samples are ready for multiomics integration.